ganz begeistert von Biontech im Moment jetzt. Sind Sie schon geimpft?
6.2. Jetzt bin ich wieder los.
Should we start?
Of course, we have everything to do.
Good morning, dear colleagues. Dear Professor Nicotera.
Yes, I'm here. Good
Great.
morning.
Good morning. Dear participants of the seminar, dear colleagues. Today we are holding Russian-German seminar on fundamental problems of neuroplasticity in diseases of the nervous system. Of course, we all know that Russia and Germany have a long history of relations in science, as well in culture, in education. And Russia always has considered scientific, technical, educational, technological cooperation with Germany is one of the highest priorities of our country, especially from the viewpoint of integration of our country into European and world scientific and technological space. We in the Russian Academy of Sciences have long-standing scientific relations with different research organizations and societies in Germany, such as Max Planck Society, Berlin, Brandenburg Academy, National Academy of Sciences Leopoldina, and of course with the Helmholtz Association. The research centers of Helmholtz Association has been for many years strategic partners of Russia in international research in different areas, such as physics, astrophysics, biology, biochemistry, earth sciences, ecology, space technology, and aviation. We carry out scientific expeditions, and we have interesting results of our joint research published in very prestigious scientific journals, and we think that our collaboration has a great potential for the future. And in this regard, collaboration in neuroscience is one of the interesting opportunities. We, as we know, we started several years ago our collaboration in this area of research, and as I remember, the first discussion on this topic was in February 2019, when we met with Professor Otmar Wissler here in Russia, in the Russian Academy of Sciences, and we expressed joint interest in developing collaboration between the Russian Academy of Sciences and the Helmholtz Association in the field of brain research. Then we performed some steps, and one of the important on this road was the visit to Moscow of a delegation of scientists from the German Center for Neurodegenerative Diseases, headed by Professor Nicotera, and holding then the Russian-German seminar on brain science and neurodegenerative diseases, here at the Presidium of the Russian Academy of Sciences. And I am very glad that despite of this difficult pandemic time, we go on with our cooperation in this topic, and we will see the result of our current collaboration today during this seminar. And of course, I welcome all participants of this seminar, and I also would like to add that on February 9, there will be this traditional day for winter meetings organized by Helmholtz Association in Moscow every year. And this year we are going to renew our agreement on scientific cooperation between the Russian Academy of Sciences and Helmholtz Association. And I hope that during this procedure on February 9, we will especially emphasize our current collaboration in neuroscience, and will refer to this today's meeting as one of the important and productive steps on this road. So welcome, and let's start our seminar.
Professor Nicoletta, your welcome speech, please.
Yes, I just would like to thank Professor Sergeyev for his introduction. I go back to our visit to Moscow just before Christmas 2 years ago, which was extremely productive, and we were very, very happy to start this kind of exchange, which unfortunately was somewhat slowed down by the corona epidemic, but we felt that there were a number of contact points between the scientists we met in Moscow at the time and the work that we do at DZNE. As you know, DZNE is focused primarily on neurodegenerative diseases, and of course our work goes from very molecular to clinical to healthcare research. I think we would be able to find several points of contact between our work and the work that is done by the scientists at the Academy and a number of others who we met in Moscow at the time. And that could be the basis for future forms of collaborations, which I think should be created. I think that this would bring us closer and would benefit both. The corona epidemic has somewhat changed a little bit the sort of the speed at which these kind of plans have been implemented. The restrictions on traveling in part are overcome by conferences like this. In part, I think the personal contact is also always very important, because you develop sort of, also personal links that help usually research to be promoted. But this should not be a hindrance. We all hope that this year, at least towards the middle and late part of this year, the situation will improve. And eventually we can then welcome, for example, our Russian colleagues back to Germany, and we can definitely start collaborative research projects. So this seminar is intended to be able to be a little partial showcase of some of the topics that could be of interest for both. And I'm particularly happy that a number of people were able to join. And without any further ado, I would like to sort of end it here and welcome all the participants, and look forward to this seminar today, which will be, I hope, the first of a series of interactions and exchanges with the Academy and with the scientists that are interested in this field of research from both sides. Thank you.
Dear Nicoletta, dear participants of the seminar, dear colleagues, let me first welcome all the participants of our joint seminar. You know, neuroscience is one of the most dynamic and rapidly progressing areas of modern world science. Major world powers such as the United States, Canada, Japan, Republic of Korea, Israel, and others are developing modern national and international brain research programs. The multi-billion-dollar investments of the leading countries in this field are due not only to the fact that the knowledge of the mechanism of the human brain and mentality is the main challenge for science in the 21st century, but also to urgent need for this research in economic and medical purposes. Germany and Russia have similar research programs and goals in this direction. It's no coincidence that the European Union launched an unprecedented 10-year flagship brain research program, the Human Brain Project, in 2013. In Russia, the National Research Program on Basic Brain Research has been already prepared and approved by the President of the country. Cooperation between our country in this field is of great interest for Russian scientists. Today, there are more than 500 diseases associated with impaired functions of the nervous system. According to various estimates, from a quarter to a third of the world population directly face these diseases. In Europe alone, 179 million people are affected by brain diseases. The cost of those diseases in developed countries exceeds the cost of treating cancer, cardiovascular diseases, and diabetes combined. In the United States, in Europe, this economic load exceeds $1 trillion American annually. A huge economic burden is also imposed by the increase in the life expectancy of the population in developing countries. And of course, the age transition, which is one of the big challenges for Russia as well. For example, according to the World Health Organization, in 2015, the global cost of age-related dementia was estimated as $818 billion American, or 1.1% of global GDP. As life expectancy continues to increase, this burden will accrue. According to the United Nations Population Fund, today the number of people over 60 is more than 12% of the total world population, and by 2050 it will increase to 22%. Our seminar today is dedicated to biomedical and clinical neuroscience. We will be able to get acquainted with the achievements of Russian and German research teams in the field of studying the mechanisms of development and advancement and the new methods for early diagnosis and treatment of socially significant brain diseases. I am very pleased to welcome Professor Nicotera, who visited Russia in December 2019, took part in the Russian-German seminar Brain Science and Neurodegenerative Diseases, made a presentation in that hall, and also visited the Scientific Center of Neurology where he got acquainted with the promising research developments of his Russian colleagues. I am absolutely sure that today's forum will be a successful continuation of the intensive and fruitful scientific cooperation between Russia and Germany. Thank you. Dear colleagues, let me start our seminar, and first to invite Professor Emrex Dusen with his presentation, Integrity, Reserve and Plasticity in Hippocampal Networks in Old Age and Alzheimer's Disease. Please, Professor.
Seems we have some problems. Emre, are you connected? Emre, are you connected? We hear you badly. There seems to be a problem. I think, let's see if I can connect with Emre Duzel, because I don't see him in the list of the connected people. Emre, can you hear me?
I'm sorry. In that case, we should continue our seminar, and maybe Professor Dusen joined us a little bit later. And
okay.
In that case, I would like to ask Professor Ryozhkin, the member of Russian Academy of Sciences, to present his report, his presentation. Please, Professor.
Good morning, Professor Sergeyev, Professor Chikhonin, Professor Nikotera. Good morning to all our colleagues and friends who are participating now in our Russian-German meeting on neurodegenerative diseases. In my presentation, I'm going predominantly to talk about three most important neurodegenerative diseases: Huntington, Parkinson's disease, and Alzheimer's disease, of course. And first of all, I'd like to thank all my colleagues and friends from the Research Center of Neurology and other institutions across Russia who contributed enormously to our joint studies and to different part of my presentations that I'm going to show today. And our group at the Research Center of Neurology has been involved in studies of genetics of neurodegenerative diseases for more than 3 decades. And we were first who were able to identify genetic structure of all main groups of neurodegenerative diseases, like Parkinson's disease, autosomal dominant, autosomal recessive cerebral ataxias, dystonias, different forms of dementia, and so on. And these studies, which have been published in many, many journals, are important not only for studying basic mechanisms of neurodegeneration and for providing DNA diagnosis and medical genetic counseling. More importantly, the identification of different mutations in the candidate genes provide a unique source for studying neuroplasticity at the early clinical and preclinical or latent stage of neurodegeneration, and I'm going to show this on different models of preclinical stage of neurodegeneration. And as all you know, the course of neurodegenerative disease is a very, very long-lasting story. And our challenge, great challenge, is to identify somehow the pathological process somewhere at this stage, relatively early stage of preclinical or early clinical stage, which is might provide the possibility to do some neuroprotective activities. And for this purpose, we need to identify a reliable preclinical marker, biomarker of neurodegeneration. And for instance, some clinical markers, like for Parkinson's disease, it's REM sleep behavior disorder. Neuroimaging marker for Parkinson's disease is hyperechogenicity of the substantia nigra, which can be seen long before the first clinical signs of Parkinson's disease. Different neurophysiological markers like color vision abnormality, and of course genetic and transcriptomic markers. And in our studies, we performed two-step population screening for Parkinson's disease risk group, for identify those people from healthy persons who might be at risk for Parkinson's disease. And first of all, on the first stage, we identified persons carrying either mutations in the Parkinsonian genes or carrying hyperechogenicity of the substantia nigra, a well-known marker of preclinical Parkinson's disease. And this was the primary risk group. Then we added several additional, not so specific, but very important physiological markers, as I showed already, and those who carried one primary marker and two or more additional markers constitute a secondary group of very high risk of Parkinson's disease. And over a 7-year period of follow-up, we were able to show that those persons carrying Parkinsonian risk are characterized by a dramatic increase in the number of different markers of neurodegeneration, while those not carrying risk factors, genetic factors, are mainly stable. Moreover, several persons already carrying those genetic or other markers have already got to be affected with Parkinson's disease. They have clinical symptoms. And none of the persons from the negative risk group affected with Parkinson's disease. And so you see that we can really identify preclinical stage of Parkinson's disease. And then, on the next step of our studies, we combined studies on patients on clinical, preclinical, and advanced clinical stage, and animal models of clinical, preclinical, and advanced clinical stage of Parkinson's disease. So we identified some transcriptome clusters of gene candidate, and very recently, just a paper published several months ago, we were able to show that both on animals in the nigra and in the striatum, we can see the dramatic decrease in the expression of the VCP protein, which is important for protein control system. And also, this is again animals, and this is patients with preclinical and early clinical stage of Parkinson's disease. This is blood cells of patients. There's the identical findings of the decrease in VCP protein. Values in containing protein. So we then reconstructed some protostatic diagram, as shown here, which demonstrate the main processes involving VCP protein and alpha-synuclein protein, which is, as you know, the main cause of molecular pathogenesis of Parkinson's disease. This is important because we can select some very promising candidate pathway or biochemical participant for target therapy for the pathogenetic studies. Concerning Alzheimer's disease, we studied, again, clinical normal people who carry mutations in several candidate genes which are responsible for Alzheimer's disease. Picalin gene, ApoE, epsilon 4 genotype, or clusterin gene. And we're able to show in a series of papers shown here that those people are characterized by the increase in the latency of cognitive working potentials, in the increased brain activation during cognitive task, this is compared to control, by the abnormality of EEG connectivity, interhemispheric connectivity, and also by the increase in the paroxysmal EEG activity on hyperventilation. And very recently, we were able to show, this paper just published also several months ago, that those clinical healthy persons who carry mutations in the Picalin gene, a strong candidate gene for Alzheimer's disease, are using the studying interhemispheric connectivity by special methods of EEG analysis like Loretta. We were able to show several brain areas in which you can see the increase of interhemispheric connectivity in the carriers of A allele versus carriers of G allele. So A allele of this Picalin gene may serve as a protective gene against age-relative cognitive decline in healthy persons. Very interesting. Moreover, this abnormality is of interhemispheric connectivity well correlated with the score of Luria memory test, and well correlated with the thickness of the corpus callosum. So you may see that we can identify some mechanism of neuroplasticity in very early stages of neurodegeneration in clinical healthy persons who are predisposed to cognitive decline upon aging. Well, another our model, also well-known model, is a neuronal culture which can be obtained from fibroblasts of patients carrying different mutations through cell reprogramming, through so-called induced pluripotent stem cell studies. And in our studies, we used dopaminergic cells obtained from patients carrying different mutations in Parkinsonian gene. We used GABAergic medium spinal neurons for Huntington disease patients, and also spinal motor neurons were obtained from patients carrying mutations for amyotrophic lateral sclerosis gene. And for Huntington, let me remind you that Huntington disease is the polyglutamine disease caused by expansion of polyglutamine repeats. And most of the authors in the past used extremely large mutant allele to express mutations at cell level because it's much easier to get pathological phenotype. But extremely large allele are not relevant to clinical practice because these patients are very rare. We were the first group in the world who were able to show that even expression of moderately or slightly moderated expanded alleles like 40 or 42 repeats are characterized by clear pathological cellular phenotype like abnormality of autophagosome formation, indentation of nucleus of neurons, and many other findings which can be seen in the brain of patients affected with Huntington disease. Moreover, using several candidate molecules of these molecules shown here, EVP, you may see that we can restore, here you may see abnormality of store-operated calcium channel inside cells. And we can restore this calcium channel by using the molecule EVP, which is a candidate molecule for neuroprotective treatment of Huntington disease. And that this paper just already accepted in press, accepted but not yet published, we will publish in 2 months very quickly. And we were able to show that this candidate molecule effectively suppress the expression of Huntington on cells obtained from patients with Huntington disease. And moreover, more interestingly, the same molecule can suppress the increased expression of STIM2 protein. STIM2 protein, this is a protein which is responsible for some intercellular, very important cellular processes and in Huntington disease. And also we can see that if we suppress the expression of this STIM protein by hairpin RNA or by pharmacological approach, we can restore calcium influx inside the cells. This is a clear candidate for new drug, which may be studied further in this cellular model. For Parkinson's disease, we studied several different genes, patients with genes for PARC2 mutation, LARC2 mutations, or GBA mutations, and obtained dopaminergic neurons. They are characterized, as shown here, by disbalance of apoptotic factors. They're characterized by the disbalance of growth factors like GDNF or BDNF, shown here. We were able to express these mutations at the 2D or 3D models, like mini organoids of brain, and we're able to show the abnormalities in the neuronal activity of multi-electrode microarray. And you may see here that's normal activity and activity with Huntington's cells and Parkinson's cells. They are completely different. This is a very convenient mechanism of studying, monitoring the effect of many drugs or the effect of the course of neurodegenerative process at this cellular model. Very interesting, very promising. And we expressed these mutations and from PARC2 and LARC2 patients, and created transcriptome map upon transcriptome analysis of this cell culture, and we're able to show for the first time that neurons carrying PARC2 mutations are characterized by the increase in the expression of many, many Parkinsonian genes, more than 60 Parkinsonian genes, probably as a result of the abnormal accumulation of these mitochondria in the cells. And neurons carrying PARC2 mutations are characterized by the metabolism predominantly via glycolysis as can be confirmed by the double increase in the expression of many glycolytic genes, which may be explained possibly by accumulation of abnormal mitochondria that cannot be effectively support oxidative phosphorylation, and neurons carrying part 2, LAC2 mutations and GBA mutations. We can see abnormal expression of the TREAM genes, which are responsible for neuroinflammation, another probably very important mechanism of early degeneration in Parkinsonian carrier of Parkinsonian mutations. These data have been published also worldwide, and they show clearly that iPSC-based models, the models based on the genetic reprogramming of somatic cells, can help us study basic mechanisms of neurodegeneration. And they are very important to develop personalized approaches to the treatment of individual patients through study of its individual neuronal culture at the cell level. It's very, very important for screening of new potential drugs. Of course, for experiment of genomic correction through CRISPR-Cas9 system, we also have such an experience. I don't have time to show in detail. And also for iPSC banking for further studies for neuroprotection, neurodegeneration, of any other very interesting, very promising direction of study. These works are in progress now and show us the potential of modern neurobiology technology in studying early preclinical latent stages of neurodegeneration. Thank you very much for your attention.
Thank you, Professor Ilarioshkin. Are there any questions to Professor Ilarioshkin?
So, regarding this induced pluripotent stem cells bank, can you just tell again just what is the idea of building this bank? Do you start with patients with some already known genetic predisposition to the disease, and then what you do? Just you take somatic cells, produce pluripotent. What is just the sequence of
well, thank you very much for your question. It's a very interesting question, very important. First of all, I'd like to say that it's very difficult to create such a bank. It's very expensive, very time-consuming and effort-consuming and so on. But it would be fantastic if we were able to create such a bank like within the national program of brain or something like this, because first of all, as I tried to show, we would be able to get inevitable sources of cells with expressing different mutations within different metabolic pathway, which can be studied at different level of our knowledge about the molecular pathogenesis of neurodegenerative diseases. Today we know about one gene. Tomorrow we'll discover another gene or another metabolic pathway, and we'll get mutations expressing different pathobiochemical changes relative to one or another pathway. It's very interesting, very fantastic perspective. And secondly, of course, we would get some sources for neurotransplantation, for neuroregenerative strategy, because such approaches are already in progress in the world, and also, by the way, in our country also as well. We have some experimental experience with animals and so on. And a lot of people affected with, for instance, Parkinson's disease, waits for new era of transplantation of dopaminic neurogen inside striatum. This can be done technically without some great problem, because it's such kind of operations already known how to do, and there is a technical assistance and so on, but we need to get some scientific basis to do that. Thank you very much again for your question.
Perhaps just one, I intercept for just one information, Professor Chegonin. I talked to Emre Dusen, and he of course mixed up the Russian time and the German time. So he assumed that he had to speak 2 hours later than he was scheduled on the program. So if you agree, I think what we could do, we could sort of make him speak either after the coffee break
which is scheduled on at 1 o'clock Russian time and at 11 o'clock, 11 o'clock our time. And we would put him perhaps immediately before Professor Lukmanov. What do you think?
I think so. It's all right. I think in 2 hours he will be in touch, and we will be happy to see him and.
Okay, very well. So I'll communicate with him and inform him. Okay, thank you very much.
Next, our colleagues, we have to continue, and our next lecturer, Professor Salmina from Krasnoyarsk, with the presentation current approaches to modeling and studying the brain neurovascular unit and neurogenic niche of neurodegeneration. Please, Professor Salmina.
Okay, thank you very much. So, yeah, can you see the presentation?
Not yet.
Okay, now it's okay?
Okay.
Okay, sorry for some technical problems. So, dear colleagues, I'd like to express my thanks to the organizers of the seminar for inviting me to explain some our activities in modeling and study of brain neurovascular unit and neurogenic niche in neurodegeneration. So thanks to Professor Petzold, we already know some important issues about the neurovascular unit. So I'd like to say that I'm from Krasnoyarsk State Medical University from Siberia, and we have some history of collaboration with Russian and foreign partners to develop translational and clinical neuroscience. And some of these studies have been focused on modeling, study, and target modulation of neurovascular unit in neurodegeneration. And also, we work closely with our clinical partners to develop new rehabilitation technologies in neurodegeneration. As to the basic neuroscience that we do at the Krasnoyarsk State Medical University, we mainly focus on aberrant neuron glia interactions and Alzheimer's disease pathology, particularly in relation to the astrocyte dysfunction. So modeling and study of blood-brain barrier and neurovascular unit and blood-brain barrier breakdown in Alzheimer's disease, and also molecular architecture of the social brain, particularly on deregulation of oxytocin release and impairment of social recognition in neurodevelopmental disorders. So you can see here our papers published in these three main directions of our activities. So I'd like to focus on pathobiochemical mechanisms of brain pathology. And as you know, in addition to some very important alterations like imbalance of excitation and inhibition, oxidative stress, induction of apoptosis and autophagy and so on, alterations in neurogenesis and aberrant activity of the neurovascular unit and the blood-brain barrier appear to be very important, either for study the molecular pathogenesis and also for searching for new molecular targets to treat neurodegeneration and neurodevelopmental disorders. So there are several approaches to model the neurovascular unit and BBB, blood-brain barrier, in vitro. Particularly, we can use different sources of the cells, like stem cells and freely obtained primary cells, like that are able to create neurospheres in vitro. And also, we can put several components together in the Transcranial model or scaffold embedding model, just to get the information about the proliferating activity and structural and functional integrity of the blood-brain barrier, and to study cell-to-cell interactions as well as release of live transmitters and several metabolites that might affect the functioning of the neurovascular unit and the blood-brain barrier. So we do some experiments with the original models of the NDU and VBB, and then we are able to study the behavioral cells in vitro, ex vivo, and even in vivo. So I'd like to show you some examples of these activities. So initially, we are very much interested in the metabolic cooperation of the cells within the neurovascular unit and blood-brain barrier, with the focus on astroglial activity that are very important glycolytically active cells able to release lactate to affect the neuronal status due to existence of the astroglial neuronal metabolic coupling, as well as to affect angiogenesis by acting at brain microvascular endothelial cells, either being uptaken by monocarboxylate transporters or acting on GPR81 receptors. So astrocytes are very interesting cells because they take part in different regulatory processes in the brain, like controlling neuronal activity and local microcirculation, as well as brain drainage and inflammation and local immune response. And of course, they're very much important as a support to stem cell maintenance, proliferation, and migration of progenitors within the conventional and non-conventional neurogenic niches. So they take part in the controlling the angiogenesis and neurogenesis in the brain. And also, you know that there is a astrocyte heterogeneity in Alzheimer's disease, because we are able to see the predominance of perivascular astroglial changes in, for example, in animals, so with the model of Alzheimer's disease. So we can see the appearance of S100 beta immunopositive astroglial cells that are the perivascular astroglial cells. And in the blood-brain barrier, in the brain microvascular endothelial cells that are surrounded by the perivascular astroglial cells, we always see the deregulated expression of tight junction proteins. So it means that probably the activated astroglial cells or deregulated astroglial cells in the perivascular area may affect the structural integrity of the blood-brain barrier. So that's why we were interested in the action of lactate that might be produced either by astrocytes or by endothelial cells and can be released through the connexin 43 hemachannels or monocarboxylate transporters, and then it may affect the activity of the brain microvascular endothelial cells. So we demonstrated that, for example, in the case of neuroinflammation that was modeled in vitro in the original BBB model, the expression of GPR81 lactate receptors was changed in endothelial microvascular endothelial cells, and we were able to show the impairment of lactate reception and transport that were very important for changing for induction of alterations in the BBB structural and functional integrity. Also, in a case of experimental Alzheimer's disease and in modeling the neurotoxic action of amyloid beta in vitro, we demonstrated that the higher astrocytes originated lactate levels resulted in GPR81 expression in brain microvascular endothelial cells. And it corresponded to the changes in the expression of CD38 in astroglial cells and activation of CD38 catalytic activity to produce cyclic ADP ribose, that is very well-known mobilizer of calcium from the endoplasmic calcium stores in astroglial cells. And it corresponded to the alterations in reactive oxygen species production, but particularly it was aggravated by the application of inhibitor CD38 expressed in astroglial cells. And also mitochondrial release was very much enhanced from the activated astroglial cells, and it was controlled by CD38 inhibitor action. So in animals with the model of Alzheimer's disease, there is increased expression of CD38 in GFAP immunopositive astrocytes, particularly in the dentate gyrus in the hippocampus area. So CD38, which is functionally connected with the connexin 43, might be involved in the activation of astroglial cells to control the neurodegeneration or some other events important in the molecular pathogenesis of Alzheimer's disease. So that's why we try to find the connection between the activity of CD38 and mitochondrial glycolytic activity of astrocytes. And we found that mitochondrial dynamics in astroglial cells as well as in brain microvascular endothelial cells was very much affected by the neurotoxic action of amyloid beta in vitro. So as you can see from the summarizing table shown in the lower panel of the slide, in astroglial cells and brain microvascular endothelial cells in the model of NVU in vitro, we found quite different changes induced by amyloid beta exposure. And the anticipated outcome from the changes in the expression of proteins controlling fusing and fission or separation of mitochondria, the activation of fission of mitochondria and reduced reactive oxygen species production, and in brain microvascular endothelial cells, it could result in the promotion of angiogenesis. So amyloid beta can induce some specific changes in the mitochondrial dynamics in microvascular endothelial cells and astrocytes that is very clearly shown in the in vitro model of the neurovascular unit. And also you might know that cell-to-cell communications within the neurovascular unit is very much important for controlling the neurogenesis in so-called neurogenic niches in our brain. Because again, there are some very interesting mechanisms that contribute to the controlling the maintenance and proliferation of neural stem cells and progenitors, and some of them are mediated by lactate release from astroglial cells that usually can be found in the close vicinity to the neurogenic niche, and that may control the activity of the vascular scaffolds within the neurovascular niche. So that's why we try to see whether it was possible to control the neurogenesis in vitro or ex vivo or in vivo by activating the astroglial cells. So to do this, we made the model of the neurovascular, of the neurogenic niche in vitro with the vascular support made from the brain microvascular endothelial cells as well as neurospheres containing some quiescent and amplifying progenitors and slowly dividing neurostem cells and astroglial cells. And then as you can see, of course, it's very evident that there are alterations in early stages of subgranular zone neurogenesis in hippocampus in Alzheimer's disease, as evident from the changes in expression of, for example, some markers of early neurogenesis or proliferation, and also usually amyloid beta oligomers can reduce the pool of stem and progenitor cells and may affect the survival and differentiation and migration of stem cells and progenitor cells. Also in vitro, the proliferative potential of cells is very much decreased in the case of amyloid beta neurotoxic action. So we decided to check the optogenetic control of astrocytes in vitro neurogenic niche model. So you can see here the schematic structure of this neurogenic niche model, and the neurospheres and astroglial cells and endothelial cells that combined within the Transcranial model, and we express the channel rhodopsin in astroglial cells under the control of GFP promoter, and then we produce the optogenetic activation of astroglial cells. So we found that photostimulation of channel rhodopsin expressing astrocytes in vitro affected the cell proliferation index and the co-culture of astrocytes with neurospheres, particularly those that were treated with amyloid beta, and also restored development of type 1 and type 2 progenitors affected by amyloid beta, and it was corresponded to changes in the proliferative activity of neurostem cells and progenitor cells. Then we use the ex vivo neurogenic niche model. We implanted the neurospheres into the hippocampus, and we checked the changes in the electrophysiological parameters in the hippocampus, and we found that stimulation of neurogenesis with the optogenetically activated astroglial cells reduced excitatory postsynaptic potentials in major neurons of hippocampus, probably due to redistribution of the pre-existing synapses, as it was shown before, or suggested before in some other papers. So that's why we concluded that photoactivation of astrocytes in vitro and ex vivo neurogenic niche model results in stimulation of cell proliferation and shifting from glial to neuronal development, so from this way to another way. And also photoactivation results in changes in lactate transport, and probably we can stimulate the neurogenesis. To check this, we performed the same experiment, but with the development of implantable artificial and optogenetically controlled neurogenic niche, just to take some neurospheres, astrocytes, endothelial cells, and neuronal cells from the mouse, and then to put into the hydrogel matrix, and then we implanted the model into the hippocampus of the mice, and we checked the appearance of these model in the hippocampus, and then we checked the expression of the markers that are responsible for different steps of proliferation and differentiation of the cells in the subgranular zone of hippocampus. So we found that expression of some early markers of neurogenesis, like radial glial cell marker 2 and PAX6 and TBR2, were increased after the photoactivation of astroglial cells before implantation of the neurogenic niche. But appearance of TBR1 as a marker of the late stage of neurogenesis was decreased. It corresponded to some reduction in the neurological alterations in the optogenetically controlled neurogenic niche implanted into the mouse. But cognitive function was probably reduced, and we decided that it might be due to a very short period before the checking the cognitive status. So this is a preliminary data that we would like to continue just to make the new protocols and regimes for optogenetic activation of astroglial cells in the in vivo implantable neurogenic niche. So as a conclusion of my short report, I'd like to show you some prospects for the development of the neurovascular unit, blood-brain barrier, and neurogenic niche in vitro, ex vivo, and in vivo studies, particularly for translational studies in neuroscience, because using this model may help us in manipulating the barrier permeability, neurogenesis, and cerebral angiogenesis. Implanted models aim to restore the cognitive reserve might be very useful in the treatment of neurodegeneration, and also some new markers of aberrant neurogenesis and cerebral angiogenesis might be available as a result of studies with reconstructed BBB, NVU, and neurogenic niche in vitro models. Thank you very much for your attention, and kind regards from Siberia, from Krasnoyarsk City.
Thank you, Professor Salmina, for your presentation. Are there any questions? Please, Professor.
Professor Salmina, thank you very much. You mentioned some defects in neuronal differentiation with your models, and there is some data in the literature discussing that probably changes in early differentiation of neurons postnatally or even in the embryonic stage might be the very first sign of late-onset neurodegeneration, which may be clinically significant many years after birth. Could you comment on this?
Thank you very much, Professor Ilorioshkin, for your question. So actually, it may correspond to the phenomenon of so-called early programming. So when we have some alterations in the prenatal or early postnatal period in brain development, then we can see the postponed alterations in the behavior or cognitive functions, and even the highest susceptibility to development of neurodegeneration, for example, several decades later. Of course, we should consider that the embryonic neurogenesis and adult neurogenesis are quite different phenomena, but probably since neurogenic niche might be affected in the developing brain, then we can expect that additional action of some factors that promote neurodegeneration, for example, accumulation of amyloid beta oligomers, might facilitate development of neurodegeneration in this population of people, this cohort of people comparing to the normal or control group. So probably it might be there some preclinical symptom. If you will be able to check the neurogenesis in vivo, actually currently it's very hard to see the neurogenesis in vivo, particularly in humans. So probably we will be able to get some information about the preclinical diagnostic tools in neurodegeneration.
Are there any questions? More questions? Thank you, Professor Salminen. I would like to invite for presentation group of Dr. Ilya Bakulin and Dr. Anton Klachkov with a presentation Modern Technologies of Neuromodulation and the Study of Brain Plasticity. Please.
Thank you very much, dear Professor Tikhonin, Professor Nicotera, dear colleagues. My name is Ilya Bakulin. I am a researcher and neurologist in the Neurorehabilitation Department of Research Center of Neurology. It is a great honor and pleasure for me to participate in this conference. I would like to talk about non-invasive neuromodulation and the study of neuroplasticity. Specifically, I will talk about transcranial brain stimulation methods such as transcranial magnetic and electrical stimulation. Transcranial magnetic stimulation, or TMS, applies magnetic pulses to the brain via a coil, and generated magnetic field induces an electrical current in the brain, which is able to elicit action potential in neurons of the targeted brain area. Transcranial electrical stimulation is a technique which delivers a weak electrical current to the scalp and can modulate membrane potential. Over the past 30 years, interest in transcranial brain stimulation has been increasing, which is demonstrated by the growing number of articles. There are two main approaches of application of transcranial brain stimulation in clinical and research practice. The online approach allows a transcranial brain stimulation protocol which is can modulate brain activity and have immediate effects on brain activity or brain functions. These effects are caused by direct interference of magnetic pulses with ongoing neuronal activity. The online approach can be used to measure of cortical excitability or plasticity, to define a new diagnostic or prognostic biomarkers, or to provide a functional brain mapping. The offline approach allows brain stimulation protocols that can induce long-term potentiation or long-term depression like neuroplasticity, and can modulate activity of stimulated area. This approach is widely used for neuromodulatory treatment in patients with different nervous system disease. To navigate to TMS with online approach can be used, for example, for functional brain mapping, for motor cortex mapping, suprathreshold transcranial magnetic stimulation of the primary motor cortex and uses muscle contractions, so-called motor evoked potentials. The effects of TMS is evaluated by EMG to detect responses induced by stimulation of each cortical site. Here you can see an example of abductor pollicis brevis muscle cortical representation in a healthy subject, and using special software we can calculate parameters of cortical maps, for example, area, weighting area, and so on. Motor cortex mapping can be used for preoperative functional brain mapping, for example, in patients with brain tumors. Transcranial magnetic stimulation is a clinically validated and FDA-approved tool for pre-surgical functional brain mapping. Here you can see an example of two brain tumors located close to the primary motor cortex with almost the same location and size, but different relation to functional motor areas. Motor cortex mapping is also used as and parameters of motor maps is widely used as a diagnostic or prognostic biomarker, especially in ALS and stroke. In patients with ALS, motor cortex mapping could help to detect the subclinical and the earlier involvement of the upper motor neuron. And motor cortex mapping allows to analyzing motor cortex plasticity or functional reorganization, for example, during neurorehabilitation. Another online approach includes the combination of transcranial magnetic stimulation under neuronavigation control and high-density EEG recording with special compatible electrodes. These techniques allows the stimulation of any given cortical areas and the recordings of local and remote electrical responses to this initial perturbation. TMS EEG provides a unique opportunity for the studying of various neurophysiological processes such as cortical excitability, effective connectivity, and neuroplasticity. One of the promising clinical applications of TMS EEG is the detection of potential for consciousness in unresponsive patients. This application is based on the idea that the consciousness requires a balance between differentiation and integration in thalamocortical networks, which is reflects as brain complexity. Using TMS EEG, we can calculate a measure of brain complexity, so-called perturbational complexity index, or PCI. This index should be high when consciousness is present and should be low whenever consciousness is lost, for example, sleep, anesthesia, coma, or chronic disorders of consciousness. Here you can see an example of a TMS EEG and PCI in healthy subject. We can see that TMS triggered responses in different cortical areas, which became active at different latencies. Compared to healthy, conscious, and wakeful individuals, the brain responses of patients with chronic disorders of consciousness are either local, suggesting loss of integration, or global but stereotypical, suggesting loss of differentiation. Therefore, we can use the PCI to discriminate between consciousness and unconsciousness and to detect a level of consciousness in patients with brain injuries. Previous studies with the validation of PCI in a late sample of patients provided an empirical cutoff 0.31, which is able to discriminate between consciousness and unconsciousness and between minimally consciousness state and patients with the vegetative state or so-called unresponsive wakefulness syndrome. In our replication study, in the correlation with the efforts of the PCI method, we performed a replication study of this method in an independently sample of patients. We found that in 12 patients with minimally consciousness state, individual PCI was higher than PCI, which is validated as cutoff. Most patients with vegetative state showed low complexity PCI values, but 4 patients provided high complexity PCI, which might suggest a covert capacity for consciousness in these patients. The ability of so-called repetitive TMS to non-invasively modulate an activity of stimulated brain regions has been translated to provide a new treatment strategies in patients with different brain diseases. Depending on the frequency of stimulation, TMS can exert excitatory or suppressive effects. According to the current evidence-based guidelines on the therapeutic use of TMS, level A, which is definitely effective, level A evidence was reached for depression, neuropathic pain, and motor stroke, and level B and C for many diseases, but however, the use of ITMS in real clinical practice is limited by the very high variability of its neurophysiological effects and very high variability of its clinical efficacy. And it was very important to provide and to develop a new approaches for more effective stimulation in real clinical practice. One of the promising approach is a more accurate targeting of stimulating coil using individual neuronavigation based, for example, on the structural anatomical MRI instead of external scalp landmarks. Another approach exploits functional neuroimaging data, for example, TESC or resting state fMRI, to personalized stimulation and to personalized selection of target for stimulation. In our lab, we are using individual structural and functional MRI to select target for stimulation in patients with stroke, multiple sclerosis, cognitive impairments, disorders of consciousness, depression, and other diseases. So, for example, we have completed the study of effectiveness of different targeting approaches in patients with resistant depression. We compared standard targeting based on anatomical MRI and personalized approach targeting an area within left dorsolateral prefrontal cortex having maximum negative functional connectivity with subgenual cingulate cortex. We observed the efficacy of both protocols, and both protocols are effective in reducing of the depression symptoms, but personalized target selection accelerated the onset of antidepressive effect. Another promising direction is the search for new stimulation protocols, for example, theta burst stimulation. Animal studies have shown that bursts repeated at the frequency of theta rhythm, 5 Hz, induces a maximal long-term potentiation. In sham-controlled study, we compared the efficacy of conventional high-frequency ITMS and theta burst stimulation in patients with progressive multiple sclerosis and severe spasticity. We observed that both protocols are effective in these patients, but compared to sham stimulation, but theta burst stimulation has a longer antispastic effect compared to high-frequency ITMS. Another field of our research is the transcranial brain stimulation in patients with chronic disorders of consciousness. In open-label, non-randomized study, we selected angular gyrus as a target for stimulation based on the hypothesis that this cortical area may serve as a very important for consciousness hub within the default mode network, and we found mild behavioral improvement, but after 10 sessions of ITMS, but only in patients with a minimally consciousness state and no effect in the patients with vegetative state or unresponsive wakefulness syndrome. Neuromodulation is widely used in patients with stroke to promote recovery and to induce neuroplastic changes. In patients with motor stroke, considering the concept of dysregulated interhemispheric interactions, the excitatory protocols are meant to be applied over lesional motor cortex and inhibitory ones over contralesional motor cortex. In sham control study, we observed that both protocols as well as its combination significantly improved motor function in patients with motor stroke. In patients with motor impairments, for example, in patients with stroke and Parkinson's disease, may be very important to assess effect of neuromodulation or neurorehabilitation on biomechanics of movements. Motion analysis lab in our center allows us to study whole body biomechanics, for example, biomechanics of movement of fingers, hand, walking, and so on in different diseases, and provide clinical and fundamental researches in this field. And patients with post-stroke hemianopia, another method of transcranial brain stimulation, transcranial direct current stimulation can be used. In a preliminary study, we observed that TDCS significantly improve visual function quality and quality of life and of these patients, and can induce neuroplastic changes in the visual cortex after stimulation. In conclusions, I would like to say that non-invasive neuromodulation is a rapidly developing field of modern neuroscience. Transcranial brain stimulation can be used for assessment of neuroplasticity and for neuromodulatory therapy, and many technological innovations could dramatically improve this field in the future. Thank you very much for your attention.
Thank you. Thank you very much. Are there any questions?
Dr. Bakurin, thank you very much. You showed us very interesting data on the result of transcranial magnetic stimulation in different conditions. But as you know, unfortunately, the extent of the effects of transcranial magnetic stimulations is relatively limited because of just physical reasons. How deeply can you get inside the brain? Not so deeply, I think. Could you please mention very briefly what might be some other physical principles of providing stimulation of the brain with other physical processes like new type of waves, laser, ultrasound, and so on, which are now in progress or in the development in the world? Just very briefly.
Thank you very much, Professor Lyoshkin, for these questions. It is very important to develop new stimulation protocols for a more deep stimulation, and I can observe several ways in this field. Firstly, it is a new coil for deep transcranial magnetic stimulation. It is ultrasound stimulation. It is a focused ultrasound stimulation. It is a very rapidly field now, and it provides opportunity to stimulate deep brain structure, for example, thalamus or other structures that are very important for disorders for consciousness, for movement disorders, and so on. And it is some approaches associated with electrical stimulation that now can be used only in animals, but I think soon can be translated in a clinical practice.
Thank you very much. I would like to invite for presentation Dr. Roman Lukmanov with a presentation brain-computer interface from theory to the clinics. Please.
Good day, dear colleagues. It's a great pleasure to speak in this auditorium. Thank you very much for inviting me. At this presentation, I will talk about main points of our own studies for brain-computer interface technology in neurorehabilitation, from fundamental aspects to routine clinical practice. In past 8 years, we made great work and published more than 30 papers about neurophysiological, neurovisualizational, neuropsychological, and of course, clinical aspects of using this technology in neurorehabilitation in healthy subjects and in post-stroke patients with hand paralysis. At first, what is brain-computer interface, or BCI, and how we use it in neurorehabilitation. In case of stroke or severe trauma, the volitional movements in hand may completely absent, but the possibility of motor imagery remains in most cases. Motor imagery or mental practice it's a rehabilitational approach to improve motor recovery in hand after or following stroke, brain injury, or another brain damage. In the process of motor imagery, our brain generates sensory motor rhythms of electroencephalogram, which we can collect and use for biofeedback to the patient. In our investigations and rehabilitation procedures, we use non-invasive, e.g. brain-computer interface, based on sensory motor rhythm recognition to realize the biofeedback provided by visual objects on monitor and hand exoskeleton movements for kinesthetic differentiation. How does it work in neuroplasticity? At the left side of the slide, you can see the scheme of possible mechanism is generated by BCI. EEG signals are used by BCI to control exoskeleton, which provides somatosensory feedback to primary motor cortex via both primary somatosensory cortex and direct thalamic input. The simultaneous activation of presynaptic inputs to M1 with postsynaptic activation leads to Hebbian potentiation, analogous to spike timing dependent plasticity. In 2013, we have published the same results of our TMS study, which showed increasing motor cortex excitability during motor imagery in BCI-trained healthy subjects. Another aim of our investigations in BCI was to discover some neurovisualizational phenomena in post-stroke patients due to the neurorehabilitation process with task, fMRI, and functional connectivity between regions of interest, intersensory motor network. In the task fMRI study, we found that using neurorehabilitation approaches with motor imagery under BCI control and biofeedback increases activity in several zones of damaged hemisphere, such as pre- and post-central gyri, insular lobe, and decreases activity both in ipsi and contralesional hemispheres, in its visual cortex and cerebellum. This dynamics of the hemodynamic response is typical for healthy subjects trained with brain-computer interface. In the process of functional connectivity analysis, we found increasing of connectivity between primary motor cortex of both hemispheres and decreasing connectivity between contralesional posterior parietal cortex and precuneus of affected hemisphere. It's a marker of normalization tendency in functional connectivity, reorganization of interaction between frontoparietal network and default mode network, as for a healthy brain. What about clinical efficacy of that approach? In our randomized controlled multicenter trial, we used Action Research Arm Test score, and here we found improvements for the BCI group in the cylinder and pinch grasping. In opposite, grasping pinch scores didn't improve in the control group, and another test results improved significantly in both groups. Later results of our study were included into several systematic reviews and meta-analysis. In addition, in this part of our study, we found that functional connectivity change correlates with Action Research Arm Test score dynamics in improvement of pinch and cylinder grasping. At the end of my presentation, I'd like to say about our results. Add the basic knowledge for the next step to develop the neurotrainer. It's a neurorehabilitation device for patients with post-stroke paralysis in hand. It's using BCI technology, robotic exoskeleton, electromyography, and functional electric stimulation with the performance of training tasks in a gaming environment. If you are interested in our results, we can share to you by email or in other channels. Thank you for your attention.
Thank you very much. Questions, comments? Please.
Dear Dr. Lukmanov, BCI can be used in patients with motor impairments, for example, with mortal stroke, for neurorehabilitation, for induced neuroplastic changes in the motor area, or for substitution of motor functions. What do you think about clinical perspectives of BCI as a method for substitution of motor functions and method for neurorehabilitation? And what do you think about invasive BCI related to clinical practice in patients with motor impairments?
Thank you for your question. About perspective of using non-invasive BCI in neurorehabilitation, at this point of knowledge and state of the art of non-invasive BCIs, it's improving of such technology to do it more convenient for outpatient using, such as dry electrodes for EEG, wireless systems, more ergonomic and less expensive devices such as exoskeletons for hand or myoelectric stimulators. About invasive BCIs, we do not use it, but we know about using in USA by several groups. How I know, it can be used about 2 years because of it can work more. And in routine using, I don't know the cases.
Okay. So, Dr. Lukmanov, my question is, can you tell us how many real BCI products are now on market in Russia or in the world, approximately, just for information?
At this point, I can buy one device in Europe. It's GTEC from Graz. and one device in Russia with medical registration. And that device, Neurotrainer, is in process of legalization in Ministry of Health.
It might be the third product in the world.
Yeah,
Oh, that's fantastic. Thank you.
with medical registration.
Yeah, sure. Yeah, just officially approved to use in clinical practice. That's very good. Thank you. More questions from our German colleagues? No questions. Thank you. We have to go on.
Next presentation is presentation of Елена Кремнева, functional MRI, mechanisms of neuroplasticity and personalized approach to the patients. Please.
Dear colleagues, dear chairman, it's a real pleasure for me to be here today, even at this online conference format, but still it's really great. And being a radiologist, so I'd like to talk about one of the most promising advanced MRI method, about functional MRI, and how we use, about our experience in Research Center of Neurology in Moscow. So first of all, our Research Center of Neurology has been at the forefront of Russian neuroscience since 1977, when the first CT scanning was performed in USSR. It was in our department, and nowadays we have 5 MRI scanners, one of them the first Prisma scanner, the first in Russia with 64 channels head coil, which is very helpful in our research, but anyway, till time we have a lot of experience in different neuroimaging areas, and the topic of my presentation is functional MRI. So functional MRI, the classical one is a task fMRI, and is based on neurovascular coupling phenomena. Then any task, any brain activity result in local neuronal activity, and via this phenomena, neurovascular coupling, we can see hemodynamic response in these local areas, and it leads to MRI signal changes. And if these changes are correlated with time course of task presentation, so the post-processing show us areas of presumed neuronal activation. So the main 3 steps are scanning, post-processing, and results, and its analysis and interpretation. But as we all know, our brain never sleeps, and it's waking and working even if we do nothing. And this is the principle for resting state fMRI. And you see here the person do nothing in the scanner, just lays still. But after taking this information, these images, the program can see if there is any regions with correlation of MRI signal changes. And again, due to this neurovascular coupling, we can see areas of spontaneous neuronal activity and the functional connectivity resulting in functional connectivity networks. And here is one of the most stable and popular one is the default mode network. Functional MRI has a broad application from fundamental study to clinical perspectives and research to neurorehabilitation. So let's have a look at some examples based on our works. One of the main topics of research in our department is functional MRI in stroke. So we try to see changes to see brain plasticity during different time points, during different rehabilitation techniques. And for example, here you see one of our original paradigm used with this plantar device. It was taken from space technology and originally is used for cosmonauts rehabilitation. And here in the Research Center of Neurology, we use it as a device for a passive fMRI paradigm as well. And we can see changes of brain activation pattern from the first day, like here, till 1 year when they're using this walking imitation paradigm, and to compare it with healthy person. And the same device, as I already told, is used for early rehabilitation itself in acute stroke unit. And here you can see results if you compare control group patients with ischemic stroke after 3 weeks from the onset without this plantar stimulation and with it. And you see the difference and quite broad activation of sensory-motor cortex, like the primary and the supplementary motor area. And it's also leads to a better outcome and better neurological prognosis in these patients. About hand activation, of course, it's a very difficult problem and severe problem for patients, especially if we're talking about right, the leading hand. And here, fMRI in a study in chronic stroke patients showed us fMRI patterns which was consistent with recovery prognosis, like from good to bad recovery. And as you can see, this red arrow is lesions hemisphere on the picture, and you can see that IPSI lesional activation correspond to better recovery prognosis than activation, than broad activation in contralesional hemisphere or in secondary and associated areas. But as always in our life, it's not so easy. For example, on the upper row, it's an example of a patient with a great IPSI lesional activation, but the patient has severe paresis. Why is it so? Because we always should remember that function never goes without structure. And we always should see the lesion localization and the tracts structure, if it's damaged or not. And if we look at this patient, you can see that corticospinal tract of lesion site cannot be reconstructed based on diffusion tangent imaging because of its severe damage. So we always try to combine different MRI techniques, and multimodality is the routine in the present neuroscience. As you can see here from our study, in chronic disorders of consciousness, the MRI protocol included standard MRI for brain structure analysis, analysis of white matter struct of structural connectivity based on diffusion tangent imaging, and functional connectivity. And you see that patients with lower degree of consciousness, like patients in vegetative state, they have the most severe brain lesions, less structure and less functional connectivity comparing to more consciousness patient. But anyway, analysis of resting state networks in this cohort led us to introduce some indices of connectome intactness and the Hertz-folded connectome intactness, and even to get just like a number to be able to differentiate minimal consciousness state and vegetative state, which is not so easy in a clinical way sometimes. And as well, you can see the difference of connectome of awake normal patient and difference in connectome matrix in patients with decreased level of consciousness. As Dr. Lukmanov has already showed you, we can train brain in healthy volunteers or in stroke patients, and this is an example of brain-computer interface study. And so the functional MRI activation is not a static one, but it's can changes. And we use it not only in patients, but in healthy volunteers as well, or for example, to study how the brain reorganized, for example, in professional sportmen. And here you can see the study in biathlete, like this is the activation of target fMRI paradigm for a person who don't know how to shoot, who is a beginner. Then the trainers, you see more broad activation, more areas influenced by the study process. And then a professional biathlete, you see a very local, a very tiny and specialized activation of the really special areas. And as we're clinicians, vice versa, we can use these knowledges back to clinic, for example, in motor recovery prognosis in multiple sclerosis patients. Here you can see they performed the active fMRI task of finger tapping, the classic one, and the analysis of neurological outcome after relapse showed that one of the group, this one, the second one, has better recovery, and if you see the fMRI pattern, you see more broad activation bilaterally and especially in ipsilesional hemisphere, like we saw in trainees in the previous slide. And in opposite, in group 1 with worse recovery, we can see that the pattern is quite similar to one with bad prognosis in stroke patients. Again, it's more activation in contralesional hemisphere. Another role of fMRI, which is rising, and Dr. Bakunin has already showed you, is fMRI for neuromodulation navigation and assessment of treatment with these methods. Why is it? Because, for example, one of the most popular targets for transcranial magnetic stimulation is dorsolateral prefrontal cortex, which is, you see from the picture, it's quite a big one and accounts at least for 2 Brodmann areas. But we need a really tiny one for individual because it's improved the result of neuromodulation. And fMRI can help us in individual targeting. For example, this one was one of our studies with TMS navigation and TMS stimulation in eating behavior, trying to change this eating behavior in metabolic syndrome. And you can see that fMRI was not only for targeting, but also we could assess the result, as well as this example of transcranial direct current stimulation in the restoration of visual fields after bilateral occipital stroke. And you can see changes of fMRI activation just after the course of 10 procedures of TDCS, and like 1 week after. Of course, the activation and effect diminished, but at least we had some improvement of function, and the fMRI can use like biomarker in this case. The most popular and probably most widely used usage of fMRI is presurgical mapping, because it's used for planning the pathway of surgical approach. It can guide the decision for course of intervention and helps to predict possible neurological deficits and to assess neuroplasticity before and after surgery. If we're talking about epilepsy surgery, in many places it has already replaced invasive WADA test with invasive to carotid arteries, which can be dangerous. If we combine all together, it can decrease operation duration and has a really high sensitivity and specificity if we're talking about primary motor cortex. As you can see from these examples, on the upper row it's primary sensory motor cortex activation. You see it's inside the peritumoral edema, and on the upper picture it's language presurgical mapping with Wernicke area, and actually we can see the whole homunculus. It's example of one of our study when the person performed 20 sequential movements of different body parts, and the different colors actually the mapping on that part. So we can do it in many ways. So to conclude, I'd like to add that functional MRI has really great perspectives, not only in neuroimaging, but in whole neuroscience. From my point of view, it helps us to color the grayscale usual day of neuroradiologists. So you see this beautiful resting state networks overlaid on 3D render of the brain. And this is my department, so thank you very much.
Thank you very much, Dr. Krimyova. Are there any questions? Please.
Thank you very much for interesting talk. You told mainly about scientific use of fMRI. What do you think about clinical use of this technique with individual analysis, not for pre-surgical mapping or for targeting of TMS, but for diagnosis or prognosis in movement disorders, in stroke or other disease? Or maybe you can describe factors that limited clinical use of this technique.
Thank you for this question. Actually, it's a keystone of the story, because most of my studies that I showed and studies that are published, usually it's a group analysis, not an individual one. And probably those of you who were involved in fMRI have seen a very old paper where one person performed the same fMRI experiment like during every day for 30 days. And you can see different results. In the same places, we see the same clusters of activation, but one day they can be smaller, another day they can be larger. So reproducibility is a little bit problematic for fMRI, and that's why it's not used so broad in clinical research. Probably not in research, as you asked, but in clinical usage. But for pre-surgical mapping, I mean the motor cortex. Usually surgeons ask for hand or for leg, or for language mapping, I mean tongue representation. So it has more reproducibility. But if we're talking about different and difficult experiments, like we saw about hippocampus in previous presentation, so the reproducibility for one person can be a problem. How to overcome this limit? It's a question probably technique developments like higher magnetic fields, more channels in the head coil, and some other factors, better shimming and adjusting, and some technical and physical stuff about MRI. It can help in future, and we see that it's helping, but not 100%.
Thank you. Thank you very much. Other question?
Yes. Dr. Krimyova, so as far as I can understand, you have many different tasks, different paradigms for functional MRI in one or another situation, like your paradigm for gait studies, as you showed us, this device and so on. Some of them are original, specially designed for one or another matter. How do you solve the problem of compatibility of this device with the MRI field, especially a high MRI field on 3 Tesla machines and so on?
So, thank you for your question. Well, for many researchers, it's a question of fantasy, because this paradigm, they can be very different, and if you don't want to spend many times thinking about it, you can just use resting state fMRI. So, but if we're talking about task fMRI, so it can use active paradigm, but for patients it's a problem because the patient with plegia, for example, they cannot move a hand or leg. So in this case, we can use passive paradigm. For example, in our early study with finger movement, another person in a scanner was moving that finger. But of course, the progress is not staying in one place, and in every hospital, in every research department, people try to make their own devices. And for example, about this walking imitation, I saw papers with very different apparatus, like something similar to bicycle, but of course it's made of paramagnetic metal and stuff, or it's plastic, or it used not electric but pneumatic mechanisms. So it can be done. We only need people to do it, and idea.
Thank you. Thank you very much. Thank you, Doctor. Dear colleagues, we practically fulfilled our program, our seminar, and I would like to ask Professor Nicoletta to conclude our seminar.
Thank you very much. I think this was a very interesting set of presentations. I can see from what has been said that there are a number of
point of contact. Definitely, I think that you will be contacted by Thomas Gasser concerning, of course, PD. We have, of course, a number of different technologies that could be combined together, from imaging to image analysis to brain stimulation. I think that there are a number of these which could be providing collaborative topics. Of course, it remains to be seen how we can maximize the possibility of this collaboration. And that's important that the scientists have contributed, whether they find partners on the other side. They would sort of exchange information, emails, and possibility then to continue this exchange. We hope that or that we can meet personally for not too long. I mean, this COVID situation is over. I don't have too much else to add. If not, if there are some questions from the participants to be addressed now, probably that's the right time.
Thank you. Professor Laryushkin, do you want to say some words in the continue of that speech?
Thank you, Professor Chichonin, Professor Nicotera. I really see just several directions of our future collaboration, which might be real just upon our complete victory over this coronavirus infection. So there are several fields which may be enhanced by our Russian and German groups' mutual enhancement of these studies, and I anticipate very much the beginning of real collaboration between our groups. Thank you. Thank you for this seminar. It's excellent exchange of opinions and ideas. Thank you very much.
Thank you very much. Dear colleagues, I think it's a really very great event in our cooperation. We started our joint activity. I would like to thank Russian team and, of course, the team of German Institute of Neurodegeneration Diseases. I think it was quite useful meeting, very useful for both sides. And I think there are a lot of possibilities and ideas to continue our experience. Of course, I would like to thank all the participants, all teams. Of course, I would like to thank the Glencoe Society. It's important. And see you later. We would like to invite our colleagues for next step of our cooperation. We are opening, and we will do our best to continue that job and to make it useful for both sides. Thank you very much.
Well, thank you also for all the nice engagement. Thank you very much.